This protocol is applicable to a wide range of screening projects aimed at developing ATPase inhibitors, either as probes for basic research or for potential clinical compounds. This method has been optimized for semi-high throughput screening applications. It's also been designed to avoid several common artifacts, and it doesn't require the handling of hazardous materials.
In our laboratory, we have been using this assay to develop new and specific non-muscle myosin 2B inhibitors for the treatment of methamphetamine use disorder. In theory, this method can easily be applied to any enzymes producing ADP. Visual demonstration helps to clarify all the important technical details.
To begin, prepare 4,500 microliters of 20 micromolar diluted actin solution for each 384 well black polystyrene microplate by diluting the concentrated actin stock solution in actin buffer. Mix the solution thoroughly by pipetting up and down 30 times to break actin filaments for reducing viscosity and heterogeneity. Then, centrifuge the solution to remove any precipitated protein present. Carefully transfer the supernatant into a clean centrifuge tube. Prepare master enzyme mix in a 15 ml conical centrifuge tube for each assay plate by combining 3,252.9 microliters of myosin buffer for the assay involving skeletal muscle myosin 2, 171.4 microliters of LDH solution, and 171.4 microliters of PK solution. Prepare master substrate mix in a 15 ml conical centrifuge tube for each plate by combining 162.1 microliters of ATP,